Hello friends, in this video today we will be talking about the next part of photosynthesis, that is the C4 plants or C4 pathway. This C4 pathway is also called as the Hatch-Slack pathway. Hatch and Slack were the two scientists who had discovered this pathway, and hence it is also named as the Hatch-Slack pathway. Now basically the C4 pathway is the pathway, or you can say an adaptation, that has been adapted by certain plants to prevent the loss because of photorespiration. We have already seen that due to photorespiration there is less amount of glucose that is produced. Instead of binding to carbon dioxide, rubisco binds to oxygen. So basically some plants like wheat, sorghum, sugarcane, euphorbia, etc. These are certain plants which have adapted a mechanism to avoid the loss that was taking place because of photorespiration. If we go to a quick revision, then we know that rubisco was behaving as oxygenase enzyme under two conditions. The two conditions was if there is increase in temperature and there is increase in concentration of oxygen. So because of these two conditions, these rubisco enzyme, instead of binding to carbon dioxide and forming the first product of the C3 cycle, it was forming another product. So to avoid these binding of rubisco to oxygenase, these plants have adapted a mechanism which is called as the C4 pathway or the Hatch-Slack pathway. Now, to avoid these two conditions, these plants, which are called as C4 plants, they have adapted certain mechanisms. So let us see those adaptations first. That what are the adaptations that has prevented the rubisco to act as oxygenase under these two conditions. The first adaptation of these plants is that they have dimorphic chloroplast. Now, dimorphic means there will be two types of chloroplast that are found in these plants. One is called as the granule chloroplast and the second is agranule chloroplast. Granule chloroplast will be the one in which the granum is present, that means thylakoids, they have arranged themselves to form the grana, whereas agranule chloroplast will be the ones in which thylakoids are absent. So if you draw a diagram, the granule chloroplast, like I said, they are going to have the thylakoids which are arranged to form the grana, and in between will be the stromal lamellae. Whereas in case of agranule chloroplast, there will be no granum present or thylakoids present. Instead, there will be just the stromal lamellae present, which is not going to help in the light. We will see it what exactly is the benefit because of the granule and agranule chloroplast. Now when I say granule chloroplast, that means the thylakoids have arranged themselves to form the grana. Basically these grana or the membrane of thylakoid is involved in non-cyclic photophosphorylation. Right? Whereas the stromal lamellae, it will not show the non-cyclic photophosphorylation process. Okay, granule chloroplast will show the non-cyclic photophosphorylation. We had studied earlier also. Non-cyclic photophosphorylation takes place in the granule membrane, where both PS1 and PS2 are situated. Whereas the agranule chloroplast does not follow the non-cyclic photophosphorylation because it has only one photosystem. So basically, these granule chloroplast in the C4 plants is present in the mesophyll cells, whereas this A granule chloroplast is situated in another type of cells present, which is called as the bundle sheath cells. Now, what are these bundle sheath cells? If you take the anatomy of these leaves of these plants, you will find that these are the epidermal cells present, upper epidermis. This is the cuticle that is protecting it. This will be the lower epidermal cells with chloroplast present. We have studied anatomy in 11th standard, right? So below, these are the epidermal cells. Right? Now, below this, there are the mesophyll cells. And you know that mesophyll cells, they are of two types. The palisade mesophyll cells and the spongy mesophyll cells. So both the type of cells are found in the mesophyll of these monocotyledonous plants. And here there will be the vascular bundle. Right? And surrounding these vascular bundle are present the large cells called as the bundle sheath cells. So these are the bundle sheath cells present. So your inner cells, that is inner side, there are vascular bundles present. That means there will be the xylem and there will be phloem present. Okay, and these vascular bundles, xylem and phloem, they are surrounded by these bundle sheath cells. So this granule chloroplast is found in these mesophyll cells. Here are present the granule chloroplast, whereas these bundle sheath cells, they show the presence of a granule chloroplast. Now, if non-cyclic photophosphorylation is taking place, right, it is going to take place in this near vicinity. Now here the cells, they are directly, this is the upper epidermis. So the upper epidermis generally comes in direct contact with the atmosphere. So on the upper layer, near to the upper epidermis, there will be more temperature. Right? So what happens is, these A granule chloroplasts, they are present in the bundle sheath cells, whereas the granule, they are present in the mesophyll cells. And in these mesophyll cells, because of the presence of granule chloroplast, there will be non-cyclic photophosphorylation taking place. So basically, the temperature is also more here, right? And the non-cyclic photophosphorylation is a part of light reaction, right? So what happens here? Light reaction is taking place in the mesophyll cells, whereas the dark reaction, or you can say the C3 cycle, here takes place in the A granule chloroplast, which are present in the bundle sheath cells. So you can say that Rubisco enzyme is going to be present in the bundle sheath cells, having the A granule chloroplast. Now, if Rubisco is present here, the high temperature is going to be present here. So basically what is happening here is that the high temperature condition is not allowed to reach up to the bundle sheath cells because of this layer of the mesophyll cells. So one condition is being fulfilled. The more temperature which is present in the mesophyll cells is being overcome by not exposing the Rubisco to that high temperature, because the Rubisco is present in the deeper cells of the leaves. Second thing is that since the granule chloroplast is present in these mesophyll cells, the oxygen also will be released in the mesophyll cells. Whereas the bundle sheath cells, then in the near vicinity there will be no oxygen present. So there will be never increased concentration of oxygen. So both the conditions are actually fulfilled, and as a result, Rubisco here is going to act as an carboxylase instead of acting as oxygenase enzyme. Again, the first adaptation is that they have dimorphic chloroplast, which is granule and agranule. Granule chloroplast is nothing but the one which has grana, and they are present in the mesophyll cells where the light reaction takes place. They are directly exposed to the atmosphere, so the increased temperature will be present here. There is no Rubisco present here because dark reaction does not occur. Instead, the Rubisco is present in the deeper cells, which are called as the bundle sheath cells, which show the presence of agranule chloroplast. These red colored cells are the bundle sheath cells, in which takes place the dark reaction or the C3 cycle. So Rubisco is present in these bundle sheath cells. High temperature present in the mesophyll cells. Oxygen released is also in the mesophyll cells because the non-cyclic photophosphorylation or the light reaction takes place in the mesophyll cells. So both the conditions, they are not affecting the Rubisco enzyme which is present in these bundle sheath cells, and as a result, Rubisco in the bundle sheath cells, instead of acting as oxygenase, it is going to act as carboxylase enzyme, which leads to the C3 cycle. The second adaptation is called as Cran's anatomy. Now, the Cran's anatomy is the picture that you see here. Okay. What happens is basically these bundle sheath cells, they are surrounded by the vascular bundles, they are surrounded by the bundle sheath cells, giving appearance of vascular bundles surrounded by the bundle sheath cells. This gives the appearance like that of a wreath. Wreath is nothing but the circular arrangement or floral arrangement. Okay, all the floral parts arranged in a circular manner. This wreath-like arrangement is called as Cran's anatomy, which is found only in the C4 plants like wheat, sorghum, sugarcane, or euphorbia. Right? So these plants, because they are showing this Cran's anatomy, wherein the vascular bundles, they are surrounded by the bundle sheath cells, they have a benefit that the high temperature condition and high concentration of oxygen does not affect the activity of Rubisco. Okay, talking about the adaptations, we will now see that how exactly is the C4 pathway conducted.